Good afternoon, everyone. This is Shuja here, welcoming you all in the ongoing annual Distinguished Lecture Series 2021, organized by the Center for Health Science and Technology, JIS Institute of Advanced Studies and Research, Kolkata, in short, JIST, JIS-ISAR. Today, we have reached the halfway mark of this lecture series to celebrate the second anniversary of our center and institute, as this is the third one of the 6 lectures scheduled throughout this month of August. Before we get into the science part, a few cliche but good information for you all. Actually, we know all good news, all good words are cliche. Our CHEST, that is the Center for Health Science and Technology, offers a master's program in medical biotechnology and bioinformatics, along with a 1-year advanced diploma program in bioinformatics and PhD program. PhD programs are primarily in different fields of medical microbiology and bioinformatics. For details, please visit our website, jisiasar.org, or you can email or call us through these contacts that you can see on screen. Our admission is going on right now. During this short span of my speech here, I have already mentioned this term bioinformatics several times. To many of you, this is still just a term that is known to have an ever-increasing significance in resolving biological problems without knowing much about it. Today, we are honored to have with us one of the pioneers of bioinformatics in India, Professor Alok Bhattacharya. Thank you so much, sir, for accepting our invitation. Without any more delay, I request my colleague Sandeep to take the stage and introduce our distinguished speaker. Just a second, throughout the inter-lecture period, as well as in the end, you are welcome to ask questions via this chat section of Zoom or in the comment section of YouTube, where we are going live right now through our YouTube channel, JIST, JIS-IASR. All those comments will automatically drop in here in the chat section. Okay, so over to you, Sandeep.
So Divya, please unmute yourself.
Thank you. Yeah. So thank you so much. It is my really great privilege to introduce today's speaker, Professor Alok Bhattacharya. So Professor Alok Bhattacharya is the recipient of Robert McNamara Fellowship from World Bank, Rockefeller Biotechnology Career Development Award, Shanti Swarup Bhatnagar Prize, and the J.C. Bose Fellowship. And he is a fellow of Indian National Science Academy, as well as Indian Academy of Sciences. He also served as one of the vice presidents of INSA. Following his PhD at Jawaharlal Nehru University, India, he trained at the National Cancer Institute and Harvard Medical School, USA. He spent a large part of his independent career at the Jawaharlal Nehru University, New Delhi, where his scientific interests were in the area of infectious diseases, biology of parasitic pathogens, and genomics. He is one of the pioneers in the field of bioinformatics and computational biology in India. He is credited with helping to set up one of the first teaching and research programs on bioinformatics and computational biology in India. So he is now the head of Department of Biology, Ashoka University, and his current research interests are rare diseases, computational genomics, and biology of pathogenic parasites. So, I mean, when we started doing PhD, we actually, I did PhD in computational biology and used to follow sir actually from very beginning of my PhD period. So, I mean, we really are fortunate to have him with us today. And today he will be talking about bioinformatics and computational biology in rare genetic disorders. So without any further delay, I am welcoming Professor Bhattacharya. Over to you, sir.
All right, thank you very much, Sandeep, and for a invitation to give a talk. There are distinguished speakers you have lined up. Seems to be a very impressive series of talks. I hope students are, and also your faculty are taking advantage of that. So without going into, just I want to share my screen. All of you can see it.
Yes, sir.
Okay, so just that Sandeep raised this issue of our starting the bioinformatics program. We started doing sequencing, I think in 89 or so, 1989. And we were one of the first groups that they were doing sequencing. And I did an EST sequencing and started doing in 92 of the parasite. And this was like manual sequencing done, and our lab probably was doing the maximum sequencing at that point in time. So obviously not that I knew, I mean, I came from chemistry, my original background, then went into biology. This concept of computation, nobody has ever talked about, The only computation thing that was going on in some theoretical biology is prey-predator models and etc. So this, but when we started generating sequencing, and at same time that NCBI was setting up the blast servers and other things at that point, and we had to send emails to get blast analysis done. It was the good old days when every sequence was sent by an email as a blast for that. You have to formulate the whole email in a way that it will get done. And the answer will come after 24 hours. So our EST analysis was done. So that's the time that we got into this whole issue about how do you figure it out what the sequences do, how to predict function, etc. And that's how we got into this whole sequence analysis. And then I think in 1996, 97, 95, we started working on a gene prediction algorithm. Those are the early days that we did. And I never knew, I mean, nobody could ever think of the fact that bioinformatics will develop into the huge field, the whole genomics will develop into huge field, and the database will go up. This advent of next generation sequencing changed also everything. So it gives me great pleasure to see the way the field has developed, not only internationally, but also in this country. And glad that we played some very small minor role in developing the field in this country. Given that, basically what I'm going to talk about is a whole area called rare genetic disorders. And I've been talking about it. I also find that people are not aware of it. Then basically I'll give you an outline in terms of what a bioinformatician or a biologist can do in this area. There's so much of problems exist that I think people should not think of other areas which are pretty saturated, and give a glimpse, not much of what we are trying to do in terms of our work in this direction. Okay. So basically, what are rare genetic disorders? These are genetic disorders, and there are 85 to 90% of them are chronic, cause degenerative disease, and they can be life-threatening. In fact, in large number of cases, they are life-threatening, and they cause extreme disability. What I mean by extreme disability is that you can, right from young age, you can be wheelchair bound or you can be bed bound. I mean, your whole movement gets affected. So what is the nature? How big is the problem? There are 6,000 of these diseases there. You can imagine that the humans have about 25,000 or roughly about 25,000 genes. Almost every gene you can find a mutation that causes some disease or other. So 6,000 diseases, not that impact. Okay, and 80% of these are rare diseases in the sense that there is some definition of a rare. In India, it is considered to be like 5 lakh patients. If you have less than 5 lakh patients in the whole country, then it's a rare. So some diseases like sickle cell anemia don't really fall within the rare, but they are also genetic disorders in that case. And they impact a very specified group. More than 50% they affect children, very low age, and some in fact affect people only in late 40s or over 40s. And diagnosis often taken years, and patients suffer from one specialist to another. Average time taken for diagnosis is about a few years. Even in USA, somebody pointed out that it could take up to 5 years to get a diagnosis. So how many numbers of such things are there around the world? There is an estimated number, which is about 300 million around the world. Okay, that's based on frequency of some of the diseases and frequency of some of the disease mutation that you see in the population. So in India, there's no really study, but we suspect that at least 70 million people, based on a global average, may be there, such diseases in this country, the patients of that disease. India being given our sociological condition of close relation marriages, we can expect that the frequency can be much higher. So, though they're called rare diseases, but many of us believe that they are not as rare as we think they are. And that's because people don't even know about it. Doctors don't know about it. High rate of misdiagnosis. And some of the confirmatory tests, the DNA sequencing tests, that can be expensive, not available to people. And then there is a cultural factor, a stigma that we have heard from many patients. They think that they just don't want to get diagnosis done because then family will get stigmatized. They will say, for example, if a boy has let's say if a brother has a disease, the sister will never get married because that's what they will say, that we don't know the children of that girl. So people don't even get diagnosis done. So we have done a small study 2-3 years back where we actually analyzed the frequency in the normal population of a disease pathogenic allele. And sort of given here in this case. And we looked at the normal population and we find that approximately 14% of the healthy Gujaratis are heterozygous for that allele. And overall Indian population is 1.5%. So we suspect at least minimum of about 40,000 such patients in the country. Whereas actual diagnosis so far is roughly about 200. So you can see that the numbers that we have can be very deceptive. And because the numbers so far are low, both the government and clinicians, common public are not aware. The problem can be at a very large scale. This underreporting, as I said, that can be a variety of problems because sometimes disease complexity is very high. There are too many exons, introns, the disease is complex, diagnosis can be very difficult. We don't have clinical experts that does it. And then the DNA sequence is not really available as a routine diagnostic procedure in many places. So the variety of reasons that there is an underreporting of such diseases. So what is the main issue in this? So main issue in diagnosis is insufficient clinical or sequencing or the facilities available for diagnosis. Essentially, a lot of these diseases you may require, let's say, an MRI for final diagnosis, etc. Many times they are not, but DNA sequence is definitely the key to do that. And but the major problem is that even if you get a diagnosis done, what do you do with such patients? Less than 5% have any kind of therapy. When I'm saying any kind of therapy means I'm not talking about cure. Even if it suppresses the progression of the disease, we call it a therapy. And quite often that is, patients want that because they want to reduce the rate of progression of the disease. And cure for such diseases are very rare. It's very, very different from even cancer. Many cancer can be, you can have a 100% remission. Okay. And large number of people live a cancer-free life for a very, very long time. Here the question is not there. Okay. The very, very few. For a few disease, some kind of therapy that are available, and one or two cases that cure is available. And the costs are very, very expensive. And the people in India cannot really, cannot access those medicines at all. Okay, USA, Europe, they have health insurance and all this thing. And India, there's no significant plan. I can tell you that large number of scientists in India, biologists, they're not aware of it. They don't want to work on this. You know, they will continue to work on cancer in spite of the fact that, you know, there are thousands of people are working on cancer all over the world. So that's another thing, that therapy is not really available. I'll just give you one example of it. This is Novartis has developed this gene therapy system for a disease called spinal muscular atrophy. And the cost of it is $2.1 million per shot. $2.1 million, that means 16 crore. So in India, some of the people are trying to, you know, use crowdfunding to get the drug. But how many people can get 16 crores for one shot? Okay. If you don't give that shot, patients are to probably die by the age of 5. Okay. And the drug companies are giving few free. For that, they are actually causing it, you know, starting a lottery. If you are a lucky parent, if you come out with a lottery, you will get shot for your child. People who fail, the children will die by the age of 3. So this is the situation why we think it's a biologically or scientifically, these are complex. There's a lot of interesting biology that people can do in it. And I'll just give you an example. There are so many cases that, you know, there is very difficult for me to actually do it. I'll just take you through some aspects of neuromuscular genetic disorder. That's something that interests me much more. So there are about 1,000 such diseases that are there. And here about 1% of the cases, some form of therapy is available. Cure, as I said, is almost minimal, not available. And as I pointed out, available therapy is extremely expensive. And because they are not short cure, it's not like taking antibiotics for 5 days and all the bacteria goes away. Okay. And then you don't have to take anything. It's not like that. And in most cases, you have to take this disease lifelong. So it can be very high. There are some drugs that cost almost $500,000 per year per patient, and you have to take it lifelong. So the choice for a scientist who is interested in this area is to find cure, and find cure in the country, in our country, so that our patients can get access. So these are categories of neuromuscular disorders. So there are a whole lot of different types of diseases that you have. Let me see. Okay, so anyway, so you can see my cursor. So you can see that there are different types of diseases that you have. These are the disease category. And these are number of genes loci that are involved in it. And you can see that there are a total of 519 different genes or loci that are involved in this, roughly about 1,000 diseases that are known under this category. So you can see that it's complex, just the neuromuscular disorder, then there are liver disorder, the blood disorder, etc. So the complexity of it, the amount of biological problems that are there, everybody can work on one disease lifetime and there won't be much competition. So what is the major complexity that is there? Because we don't understand the link between genotype and phenotype. That we know where the mutations are. We know which genes they are, but we don't know how that mutation leads to the disease phenotype. Okay, and that is the problem with many neuromuscular disorders. Okay, that it's not that we don't know for every other diseases, but we don't know for a large number. A few diseases like Duchenne muscular dystrophy, we understand, but we don't understand fully. Unlike hematopoietic diseases, sickle cell, this, that, the biology has been worked out very well. So there the genotype, phenotype relationships are not. But neuromuscular disorders are the one category where a lot of such studies are very important. Some examples, let's say that there's a disease called spastic paraplegia that causes problems in a bone or muscle weaknesses and stiffness in it. Okay, and it causes progressive weakness and spasticity of the legs. And if you really look at it, the number of genes involved in this is so many of them, so many of them. And all of them lead to the same phenotype, spastic paraplegia. So obviously the question is how so many genes are coming in, what gene is doing, which mutation in which gene would cause anything else. Many of these genes are unimportant genes, housekeeping genes, etc. So this is an example when multiple genes can cause the same phenotype, like the previous one, spastic paraplegia. Limb-girdle muscular dystrophy is a fairly common muscular dystrophy seen in India. And there are large number of different types of it, from 2A to all the way to 2O. And you can see that there are a whole lot of different genes that are involved in it. Okay, and if not in genes, there are different kinds of proteins, sarcoglycan family, gamma, alpha, beta, delta, etc. And mutation in gene cause essentially the same phenotype. Okay, so you can see that again, like the previous slide, here also you can see how multiple genes are involved in similar kind of function in a way that mutations in this gene would cause a similar phenotype. So how do you correlate genotype with phenotype in this case? The genes are different, everything is different. Some of them are even in different parts. For example, in this case, proteinomethyl mannose transferase is a different enzyme. It's involved in mannose transferase, etc. So you can see that how it is. On the other hand, we also see different mutation in the same gene can cause multiple diseases. Okay. So, for example, all of you may be familiar with the iodine receptor gene, an important gene and transport system in the cell. different mutation in this would cause the different types of muscle disorders, and these are listed in there. So in the same gene, different mutation, different disease. Again, how different mutations cause different disease, the relationships are not very clearly. If it is single pathway where it is involved, you can think of it. You can think of a fact that if there are multiple domains of a protein, each one has a different function, mutation in each one would cause different disease. You can think of it, but there are so many, and it doesn't have so many domains also. How does that happen? So there are large number of genes that are where the mutations have been mapped for a muscle disorder. And if you really look at these genes, many of them have known to have muscle specific function. Okay. And so some of them are I've listed here. Myosin heavy chain 8, for example, myosin heavy chain 6, one of them causes laying myopathy. And then few of these, which are very clearly muscle proteins, you know the function of them, like alpha-actinin 2, you know their function, etc. And they are this. But you also have mutation in this set of genes, which are nothing to do only, I mean, they are involved in a large number of different cell types. Okay. And so why mutation in this would cause muscle-related function is not really clear. And I'll talk about, we have done a bit of work on the GNE. It's a sialic acid biosynthetic enzyme. Okay. And sialic acid is present in almost all tissues. In fact, they're very high in brain, liver, and it produces a fairly high quantity in salivary glands. Okay. But mutation in this gene causes only skeletal muscle defect. And we just don't have any idea why. So what are the priorities that we think is for the nation, and something that I want all of you to think seriously about? So we have to identify all patients. And we have to improve diagnostic methods and other things. How do we do that? We have to find cure for diseases, some treatment options, how to slow down the progression, etc., etc. And then all patients must have access to drugs, and the drugs should be made in affordable price. Like COVID vaccines, this should also be made at affordable, and this should be a national priority. And then also something that I'll not deal with later, since all the patients have a severe disability, they need some assistive devices that make their life better. And an engineering place people can understand. For example, if you have a very nice wheelchair that can climb stairs, it could be very useful. And they will become more mobile. So why therapies are not being developed in India? I don't have to really go in detail. All of you know there are problems for rare diseases because there are very few diagnosed patients. There's a lack of interest because people think they're rare. But as I pointed out, they're not that rare. The overall number of rare patients in these are much higher. Then other thing is that, and this is something that I feel important, is that our understanding of the pathobiology of the diseases are not there, really. And we're not, very few youngsters wants to work on this area. And I've talked to hundreds and thousands of young kids as well as young faculty. Everybody has this, majority wants to solve cancer problem, which is, I don't think they can. And then others want to solve tuberculosis problem or this or environment problems. Nobody thinks of these diseases as a potential area of interest. And the reason why I think it is important for me to point out here is that they are great biological problems. And anything you do in this area would benefit patients. And that's something that lack of awareness about researchers, politicians, and how the effort is happening elsewhere. Unfortunately, not in India, but Europe and America, last few years, hundreds of new startups have come, or even old companies like Novartis and others got in. And almost every company has a rare disease division where they are actually working on developing new therapies, etc. for that. Again, we are going to be way behind them. Okay, they're going to lead us. They're going to develop the IP. They're going to produce those drugs, and maybe they will make some Indian company to manufacture them. But the whole IP and whole technology will be developed. So where does the computation biologist come in? So obviously they come in for diagnosis as well as cure, finding therapies for that. And I'm sorry to say, whatever everybody else says, as a country, we have a dismal record of finding new therapies. So we haven't really found a heck of a lot of new therapy molecules or new therapeutic systems in the country. The whole development in the area of gene therapy, gene editing, everything is happening abroad. There are minuscule efforts in this country. Most of the efforts have been towards cancer and lifestyle diseases. Our record against infectious is also not much better, other than coronavirus, because the government support for that has increased quite a bit. We haven't really come up with so far a new drug for tuberculosis, for that matter, for anything else. And for this genetic disorders, neuromuscular genetic disorders. And there is we have to think of new ways of doing it, and how, we clinicians, patients, regulators, industry, they come together. There's a new paradigm today where patients are involved in drug discovery, and patients-based regulatory approvals are being. See, the important thing what is happening is that now patients kind of influence the FDA to provide clearances for drugs. And that's a very important thing, because getting clearance for any other drug is very difficult. For genetic disorders, patient groups are very strong. In America, they play a very important role to do it. There's a kind of new. There's also a new way of doing clinical trials where patient reported outcomes. That's a new approach. Plays a very important role to do that. I also want to point out, and that's something I don't know how many of you have done. I doubt anybody has done. You should look at, if you're thinking of translation, you should look at, or you're thinking of drugs, particularly if you're thinking of drugs, you should think of what are the rules that govern, clearance of drugs. So we have a new clinical trial guidelines that has come up in March 2020. They've given some concession to rare or orphan genetic disorders. Those are very important because our translation must be embedded into the, the whole aspects of regulation. And this aspect, something we scientists tend to neglect. Even I used to neglect. I didn't know anything about it. The last few years I've been into this, so I look at it and then I see that how important it is to design your work, plan your work in a way that you can eventually get clearance. So what are the possible therapeutic molecules and cure? You can have small molecules and metabolites like any other drug. You can have small RNA molecules. It could be siRNA, shRNA, miRNA, other regulatory RNAs, large RNA molecules like messenger RNAs, and then large biological like genes and proteins. Then you have a viral-based gene delivery systems also, and non-viral gene delivery systems. These are packages to deliver genes to the right tissues. That is there. So there can be a whole lot of different ways to do that, and gene editing also comes into. this whole area. So if we just simply, I kind of go over this biology part, there may be some students there. So what happens that there's a DNA and there's a mutation somewhere in a gene and it undergoes transcription. Sometimes this can get affected. The mutation can be in such a location that transcription itself can get affected. Okay. Let's say the transcription is not affected. In that case, normal level of RNA is made, messenger RNA. That gets translated. Now, again, the mutation could cause some problem here. Okay, it doesn't get translated properly, or the translated proteins don't fold properly. Okay, and even if protein is folded somewhat or didn't fold, so if it didn't fold properly, it can get degraded. The mutant protein could be less metabolically active, okay, and they can produce less metabolite. So, so you at every step you can have a potential therapy. For example, if transcription is affected, you can try increasing the level of transcription, okay, or you can actually introduce a wild type gene here, okay, or you can do DNA editing so that you make the right kind of protein. Okay, so you can also have, at this level, like the coronavirus vaccine, the Moderna or the Pfizer vaccine, you can actually introduce messenger RNA, which is based on the wild type protein, so that can be done. You can actually interfere here if the folding is a problem. You can have chaperones that will cause folding. If the enzyme activity is low, you can design a small molecule that will activate the enzyme, or you can have a metabolic supplementation. So you can have therapeutic levels all across the standard biology landscape that we know, from genes all the way to metabolites. So I sort of go over many of them. These are the various patient things that can happen and that can influence patient phenotype. And that has to be worked out for every disease. It could be a folding problem that creates problem. It could be protein interaction, can be other thing that can be influenced by mutation. As I say, gene expression. Sometimes mutant protein is mislocalized in the cell. That is, it can increase genome instability. There are examples for all of them. Interfere with signal transduction pathway, autophagy, apoptosis are affected. The activity of the protein gets reduced, degradation, etc. So the patient phenotype can be influenced by a large number of different processes and mechanisms that is caused by individual mutation. So let's look at small molecule-based therapy. What are the various possibilities that? So you could have metabolic supplementation. So let's say that because finally the enzyme is less active, and there are certain metabolites which are less or something. So you could actually give supplementation of metabolites in a way that that will augment the level of it, and that's also certainly done for a given disease. In some cases, what happens is that because of the inactivity or reduced activity of it, some toxic metabolites like phenylketonuria, the toxic metabolites can accumulate. So you design your diet, other things that phenylalanine is not present, etc. So the specific design drug can also, food can also help in that. So in some cases, you can inhibit a toxic proteins. Mutation can cause toxicity, proteins that can be toxic, and you can inhibit the protein and activate that. That can be the interesting part, and something that a good bioinformatician should work on it, and I will dwell with it. that you can activate a functionally impaired mutated proteins by small molecules. That means that mutation reduces the activity and you increase the activity by small molecules. You can inhibit unfolded protein response by allowing protein to respond properly, or the pathway for unfolded protein response is blocked. And chemical chaperones can help in mutated protein. You can stimulate pathways to bypass the toxic effect of mutated proteins. You can do that also, so that the mutated protein is not utilized. And you can stimulate transcription translation. Sometimes mutated proteins is, for example, it is less active. But if you increase the transcription translation, maybe you produce more proteins, and that can be this. Or if your transcription is affected or translation is affected, then you can have small molecule that will activate. So these are the possible do it. I give example from a little bit of work that we have done for last few years. For example, let's look at metabolic supplementation to augment the little settlement. So our idea, and I'm sure you know, that final concentration of a metabolite sum total of all the activities, number of pathways, involved with both synthesis as well as in degradation. Everything together, the whole is a network, and that leads to final concentration of a metabolite. And this is sum total of it. One way to determine metabolite to be used and the concentration, how to really come out with a therapy, is to computation and model the complete system. You can model the complete system and then determine the effect of mutation and the effect of supplementation, how much to supplement it. I just give you an example from our work. This is in GNE myopathy. I told you GNE gene is involved, it's a bifunctional enzyme involved in sialic acid bisulfite pathway, and this sort of outlines almost 35 genes are involved, 2 bypass steps are there, and degradation, all sorts of things, scavenging, everything is there in this state. So overall regulation, finally, what you are measuring is sialic acid levels or sialylated glycoprotein levels. So if there is a mutation in GNE, it can reduce the activity of GNE, it can produce less this thing, but there were other pathways that can sometimes compensate. And if those pathways are more in some patients, they may see lesser phenotype. In some cases, people see much more intense phenotype, depending upon the whole system. So what we have done is that we have set up differential equations for a large number of this, wherever we could get hold of kinetic parameters. And we have actually, I'm not going to detail in it, but you can actually model the whole thing and ask the question is that if I provide some of the intermediate metabolites like anastyl manosamine, what will be the, for example, the final sialylated levels are. So this is the way we have done it. This is just few simulation. I just give you an example. It's much more complicated than this, but this is the kind of approach that one can actually take to understand how we can actually design a proper metabolite and what concentration that metabolite has to be. Okay, so that's kind of bioinformatics or computation biology that can be done. That total, you can do a stochastic level modeling, or you can do a standard differential equation based modeling. So the other important point, I feel that many people can do it. A bioinformatician, particularly structured bioinformatician, can do it. Is to try to determine mutation that find small molecules that are activators. In fact, We know that mutation can cause quite often loss of activity or function. It can even cause, for example, the protein-protein interactions or whatever it is, the localization. One way to find a possible drug is to design or identify a molecule. It could actually that restores the function. And that can be possible if you think the mutation is causing small change in conformation and that causes change in activity. Mind you that there are not many null mutations that are there. Generally, the null mutations, that is a mutation that will completely inactivate, and not that often seen in it. So, okay. So most of the mutations are away from active site and causes a small change in the activity. So, as I said, one way to find a possible drug is to design or identify a small molecule that can restore the function. You know, people are, as all of you know, that more interested, or they're more familiar with finding inhibitors. Easy to say, I have an active site or whatever site I have, and I can design a molecule that fit into that cavity of the active site. That's fine. But how do you design activators? Okay, so allosteric sites are not that simple to find. Even if you find it, how do you design molecules that will do it? You need to have molecules binding to some other site, and it will actually stay. So this is a bioinformatics challenge that needs to be solved. Okay, and this is one area that a lot of work can be done. Mutation also hamper proper folding of the molecule. You know, a lot of simulations can be done to see how the mutations affect for folding and how to design small molecules that can actually prevent, which will act like chemical chaperones and will prevent misfolding of it. So, and those again can be done mostly by simulation. And these are useful approach to find drugs. Small molecules can be for transcription, translation, regulation. For example, nonsense mutation can cause truncation of proteins. Okay, and that will actually abrogate all the function. So one way to do it is to do a nonsense suppression. Okay, so you design a small molecule that can suppress the effect of mutation and allow read through of the translation. And already there's one example which has got FDA approval and now in the clinics called ataluren. It's not a great efficient molecule, but there's a lot of potential to design such molecules. So small molecules can also potentially interact with transcription translation, especially increase or decrease production. Sometimes mutant proteins production can be toxic. So sometimes you want to decrease the production of a protein. And this can also be played around and new molecules can be developed. In some of the diseases, for example, HDAC inhibitors have been found to be useful in therapy. Then there are therapeutic RNAs, designing as antisense RNAs that can do exon skipping, that is the exons containing a nonsense mutation. You skip it, okay, during splicing, and then what happens is that you produce a protein without that particular exon, but quite often this exon absence does not cause too much problem in the function. Otherwise, you can see that antisense mutation will cause complete truncation of the protein. So this kind of thing, microRNAs can be designed that can fine-tune the expression of genes and systems that can be done. There is a tremendous scope for work in this area. Then there are therapeutic proteins, many disease-specific lysosomal storage disorders. Enzymes are, purified enzymes are actually used as a therapeutic molecule. There are a number of issues involved, stability of it, because there's one thing that stability in bottles as well as stability inside the human body is important. Longer the protein stays, more effective it is. The uptake of the proteins, how it is distributed, cost of production, immunogenicity, those are all important issues that can all be handled partly through computation method. But more interestingly, there's some work going on, and this is really a forefront area in designing protein mimics. That means you design small molecules that can perform the same function as the enzyme, as a peptide. So, designing such molecules, people have done that for antibodies, people are doing it for enzymes. You can even design a small peptide that performs such function, or design peptide small molecule complexes or RNA complexes. So there's a lot of scope for designing here synthetic biology, and a lot of computation synthetic biology approaches can be used for that. This is one area that nobody in this country, I mean, most of this area, nobody in this country is doing. So you will be the one that will pioneer if you start somewhere. So how bioinformatics is used in gene therapy, there is a tremendous amount of scope in it. Generally, gene therapy involves use of wild type gene, introduction into a body. You allow a wild type gene expression to happen. You can replace mutant gene with a wild type gene, or you can actually also correct the wild mutation in it. And different vectors are used to actually deliver these genes. And though the most of the trials that are going on in the world are based on viral code vector, but there's a lot of interest and work going on in non-viral organized structures as a delivery system. Now, the viral design requires a lot of computation bioinformatics in it. You want to design something which is small in size. For example, if you take adeno-associated virus, can you reduce the size by computation? Can you make it less immunogenic by looking at all immunogenic sites? Okay. Can you make it more specific for muscles, for example, to be picked up, etc. And then how do you express it? Express this protein using promoters. A lot of promoters are already patented, so it's important for us to come out with our own promoters, which are tissue specific. Let's say we want muscle specific promoters to express a protein in muscle cells. We need to have promoter design, and there is a lot of scope for good design principles in this area. So the other area is how much time I have?
Sir, you please continue. Don't worry about the time.
Don't worry about the time. Okay, so the other area that I think is important for computation bioinformatics is looking at the genomics and the sequence part of it, how the distribution of pathogenic allele happens in population, estimating patient burden, the kind of work that we did by looking at pathogenic alleles, how identification of other alleles that influence penetration of the pathogenic allele. These are very important. What are the genes that can affect pathogenic allele? How do you predict pathogenic effect of a novel and unknown mutation? There are some softwares, but that 7, 8 different softwares that talk about if you, let's say, if you replace one amino acid with another amino acid because of mutation, what will do? But a lot of these depends upon the context, the protein, etc. So a good tool, I think AI-based tools could be a tremendous useful looking at this. And then we need tools for rapid diagnosis. This is something I keep talking about it. There is some work going on by which facial features, sometimes mutation in an unrelated gene can change the facial features. They may have nothing to do with face. They may be affecting leg muscles, but facial features could actually could be an early diagnosis system. So there are a tremendous amount of possibility in terms of how we can apply different computation tools to do that. And one of the major questions that clinicians ask us, patients ask us once they get diagnosis is that what is our prognosis? How bad will be? How soon we'll become wheelchair bound? And this is something that again requires a tremendous amount of work. I think, and there's a need to look at not only mutation, other clinical data, etc. Do that. I know of siblings, two siblings, for example, I know where one of them is wheelchair bound, other is able to walk around pretty well. So how does that happen? And what influences the phenotype? These are questions that we need to answer, and all of this, a lot of these things can be done computation. So I just talked about a little bit about evolution and spread, that is how this disease is spread through the population. This is another area of computation biology or bioinformatics that is an interesting, the evolution of diseases, how a mutation, a pathogenic mutation survive in a population in spite of natural selection. So these kind of questions are something that we need to answer. And a lot of research can go into understanding this sort of thing. For example, many Mendelian diseases are caused by single highly penetrating allele, for example. A simple model for Parkinson mutation that led to a Mendelian disease is that the frequencies reflect equilibrium between introduction and elimination, because the elimination can happen because of purifying selection. So one way the mutations are being introduced, other way they are being removed. So there's a mutation selection balance theory or hypothesis. So the lot of things happen. There are stochastic processes, that the random drift that happens, the demographic changes that happens, the genotype change. Those are all interesting questions that we need to. And also the genetic interaction plays a very important role. In his disease, mutation penetration. Okay, so all these things I think are important. And understanding this can be very good. For example, if gene's mutation may be rescued by another mutation in the same gene, and though it hasn't been seen too much, but it has been seen in genetic mutations also. For example, there was a study done where 566,000 human genome sequences were analyzed, and they found 12 people, only 12 people out of 566,000, but they found 12 people who have mutations which cause severe gene, and they're homozygous for that. Okay, but these 12 people were perfectly healthy. Now, the question is what are the other alleles that are present that is actually suppressing the effect of the mutant? If you can find that, we will be able to find a cure for those diseases. So a modifier locus elsewhere in the genome that modulates the severity of the disease and also the penetrance of the disease allele. It's a very useful one. And developing methods that we can find these things can be very really useful for finding drugs for this. So in conclusion, I will say that rare genetic disorders offer an excellent system for application of biology and bioinformatics. Biology, uh, computation biology and bioinformatics, these provide excellent system for basic as well as applied studies. There's a tremendous scope to use tools for developing uh, new therapies, useful therapies. This can also help us to understand burden of disease alleles, how it spreads to the population, how to reduce the overall burden. More importantly, we need to increase the awareness of all scientists so that people are aware of that there is a whole area of research that can be done, which is not going to be duplication of what is going on around the world. It will be new and unique. And I can tell you that uh, phenotypic differences, effect of a mutation on phenotype, that varies between population and population. Even the mutations keep changing. So this can be very India-centric and I would say top of the line research in biology, which can lead to opportunities for patients. Eventually, a patient, if he knows that he will never become wheelchair bound because there is a medicine, that can bring a lot of smiles in their face. Thank you very much for listening to me and giving time, and particularly listening to my new area. And I'm more like an activist here, trying to sell this whole area of rare genetic disorders. And I want people to work on this area. Thank you again. And thank you for inviting me for this.
Thank you, sir, for this very important talk. I mean, focusing on the field of rare diseases, which is becoming more complex and heterogeneous. And I think this landscape of rare disease is really constantly changing with time, and more different reports are coming showing the complexity of disease and the role of bioinformaticians or computational biologists in this disease. This is really eye-opening talk for all of us. In India, actually, I think in India, population-wise, rare genetic disease, I mean, total number will be pretty huge. I mean, if we go and check and screen them, so in different groups, so we can find different genetic diseases, and as a group, I think they will not be rare. I mean, and I don't know. I mean, I heard that Ministry of Health and Family Welfare, they actually were looking for some national policy for the treatment of rare diseases in 2017. So, I mean, these type of policies, I think, will be very important for our country, because as you mentioned that the treatment and other things are pretty expensive, and different options are available. So, so I'm just starting the question answer. So if you just enlighten us about these national policies and these things which is going on from the health ministry or something like that, if we have any national policy on these rare genetic diseases.
So, Bob, I think it's an eyewash. I know about this policy a lot. There's nothing that they're not serious, because actually the patients went to court because this treatment is so expensive that nobody can do it. So the patients went to court, and the court asked government to come out with a policy. And government came out with some policy without putting any money in it. So 17, they came out with the policy, 18, they withdrew it. This year, again, they came out with a policy because there's a problem. And there's no money allocated in that policy. I mean, how do you do anything? Where will people find money? And we have been, I've been writing and telling them that put money in R&D, put money in production. Let's say gene therapy, you want to do it, let's make centers which have gene therapy, make companies, ask them to set up an AAV production facility, which are very complicated. So this is one of those things where the government comes out with policy with no commitment at all from their side. Because court asked them, so they now tell court that we have a policy. Now where the money is going to come from, they will have a crowdsourcing. Imagine, and all that you need is a few hundred crores to set up, let's say, production center or this thing. Gene therapy is going to be eventually the most important thing. Small molecules will also help. And India can do it. We can sell it to the world once we find it. There's a lot of possibility. I mean, I can tell you that it's not that simple to come out with a drug for cancer, because the whole gamut of trials, everything is not that simple. Whereas if you want to do trial for rare genetic disorder, you need only few patients. And patients are willing to enroll because they want drug. So it's much more, and government has given at least few incentive for, the restrictions are not that much, the rigorousness is less in clearance for rare genetic drugs. So I think this is one of those cases where they just want to say that, okay, we have a policy, court asks us to do a policy, we have a policy, we don't care what the policy is.
Okay, I understand, sir. So there are few questions from the participants. So thanking you for this nice talk. And from Iftikhar, thank you, sir, for your wonderful talk. My question, is it correct to say that through thorough documentation of the true extent of genetic variations and burden of rare genetic diseases in Indian scenario is lacking?
Yeah, it is really lacking. There's supposed to be a registry coming up. ICMR is funding it, but there's only few hospitals they are involved in it. Now the question is that how do I find all the patients of this thing? A lot of patients, villages, this, that. People are dying at early age. They don't go to doctors. Okay. If somebody, let's say, have a problem in walking, how would you, and that could be because they have a genetic problem. So you need to go through the whole process of diagnosis, and that could involve even DNA sequencing. Or the government could have a mechanism by which they can screen almost all the young people. Okay. And the tools can be developed by which you can screen all the young people. Try to figure out how many actually such persons are there. Because one thing, if I know that I carry a pathogenic allele in a heterozygous situation, then I'll be careful even when I'm getting married, who will be my wife, and even a baby that is born. I don't want my baby to come up with such a this thing. So, one can plan things. We can even prevent birth of a lot of these. So the whole database diagnosis, this thing is as important as developing the therapy for it. That's something that.
So another question from Rachana. Thank you very much, sir, for this wonderful talk. Accurate patients' information are much important for studying these diseases. Sir, can you please tell if any such database along with patient information are available for India?
There are few publications that has come out. As I said, ICMR is doing a registry of a few diseases for a few centers. They are not collecting data from the country. It's only a few big hospitals that are collecting. There are some publications that has come out where they talk about what kind of patient they see. Number can be, sometimes you get a very interesting results out of it. For example, there's a study done, they found that GNE myopathy is this disease which causes skeletal muscle problem in young adults. They eventually, by the age of 30, 35, they become wheelchair bound. They found that a group of Rajasthani patients they found a mutation. And that mutation was seen only in the gypsy population of Europe. Okay. So gypsy population of Europe and a Rajasthani group of patients share the same mutation. And this mutation not seen anywhere else in the world. So sometimes some of these studies can lead to the fact that, and there's been a lot of talk about where does this gypsies, they're called Roma gypsies in Europe, where did they come from? Okay. And that you can see that, there has been either the Roma gypsies were migrated out of Rajasthan or the Rajasthanis came from Europe from that. Some of the very interesting things can emerge. There are publications, but not that many. As you said, that there are very few people who work on this.
Exactly, sir. So another question. Thanks so much, sir, for such an insightful talk about an otherwise neglected area of research. In microbial world, we often see anti-virulence genes where pathogen evolves through pseudogenization and gene loss. In humans, if a gene is associated with, say, a type of muscular dystrophy and there is a mutant variant causing the disease, are there any scenarios where we find such a disease causing variant naturally accumulates a truncation mutation in the next generations, thereby avoiding the hereditary impact of the disease?
So, that DMD, I mean, I didn't catch this whole thing, whether in the next generation, what happens is that, so let's say in the muscular dystrophy, look at it, it happens in male and the females can be carrier of that. Again, there's only certain percentage, not all offsprings will get that disease. So that way you can do that. But on the other hand, this is possible that in certain combination, for some disease, it is possible that the mother and father can bring in as a set of genes that father's pathogenic genes may be suppressed by the mother has a gene. So in order to get that kind of data, we need to do a lot more larger study to do that. Okay. Exactly. For example, if I study families where there is a carrier for some gene is, some disease is there, and we see that how the people in their family married and which ones, children from which marriages landed up with the disease and which one did not. Similar kind of genetic studies could be very, very useful to do that. But then you have to plan the studies and you have to do the studies in a larger scale. Problem is that large-scale genomic or genetic studies are just beginning to underway in India. I mean, already lot of data is available elsewhere. For example, there's a company called 23andMe in America. They have millions and millions of American DNA sequence data, where people actually give their DNA and get the sequencing done for about $1,000 or $2,000, and they want a prediction from that. But as a result, the company has such enormous amount of data. And in India, such data is lacking. The Genome India project has just started, and that too only 10,000 people sequence. So we have everywhere we lack. You want to do bioinformatics, you need data, and that's something that's not happening too much. But that's a good idea that, and possibly one can study to see how this can be done by looking into families. I mean,
yes, sir. Data is the new power source. I mean, that will be the new way of power. If you data, you have the power. So I have just a small question. So as you showed the different like small molecules and other kind of strategies for treatment of these kind of rare genetic different rare genetic diseases like neurological monogenic diseases and others, and you showed that the metabolic supplementation is a way for these kind of diseases. Whether we can design different probiotic cocktails, actually now microbes, they also can actually supplement different metabolites and other small molecules. So whether that is also possible for those kind of individuals?
I think Sandeep is a very good question, and I think this is one area that has not been studied much with respect to genetic disorders. How does the, for example, gut microbiome play or oral microbiome role in the whole thing? Is though there are enough data to suggest that muscle mass of muscle is related to what kind of microbiome one has. So the microbiome relation with muscle mass is something, and so I personally believe that there is a possibility that manipulation of gut microbiome can help in many of these genetic disorders. But we need to study this thing in great detail. In terms of ability to release some of the metabolites which are missing because of mutation, that's also possible. For example, GNE myopathy, sialic acid levels, this, that. There are microbes that make sialic acid, and studying them and seeing how much they will actually produce, whether they can actually take care of the deficiency that is there. That's something that is can be done. This is an area that can be studied, and I think it is worth studying it. And one needs to do it. This is something I'm thinking of mentioning, but then I decided that I'll stick to bioinformatics only.
Okay.
So I didn't actually see. It's good that you pointed that out. But this is a
Yes,
very good area to get into.
yes, sir. As you always used to say that not only the microbes but other protozoan, helminths,
Yeah.
all these things present in
They are
Indian
all
gut,
there
and
in the
that,
gut, They're all there in the gut.
that will be really fascinating to look at. And there is no more questions. And thank you again, sir, for accepting the invitation. And I am thanking you for delivering such an illuminating talk today. On behalf of Chest GIS ISR family, I convey my sincere gratitude for your time. Thank you so much, sir.
Okay, thank you very
Okay.
much.
So we are now leaving. Thank you all.